Okay, so what we're mostly going to be talking about are a little bit about taxonomy. Taxonomy is how things get classified in biology. When they first came out with the system of taxonomy, what was it based on? What decided what got put in what group? Nobody? Think about this for a minute. The guy who came up with the current system of classification was a guy named Carolus Linnaeus. He's a botanist. And there were lots of reasons to come up with a standard system. One is scientists from all over the world can be studying the same things, writing about them. And let's say Draven was a botanist in Asia, and Adam's a botanist in Canada. They could be studying the same thing, reading each other's papers, and not realize they were the same thing because they were called different things in different places. So it was important to have a standard naming system worldwide. That's one of the reasons. But Linnaeus is a long time ago. Before we knew about genetics and stuff. So when Linnaeus came up with this system, things were mostly classified based on this. What's morphology? Shape. What they were like. Their structure. They're not so much their function even, just their shape. What they look like. That's how they got classified back a long time ago. Today, that's not really how things get classified anymore. Today, the taxonomy system is phylogenetically based. And that's a big word that's going to show up a lot in this class, in these sessions and in regular class. Anybody want to take a shot at what it means, phylogenetically? That shower makes a big difference for y'all. It means based on evolution and genetics. Things are classified today based on how they're genetically related and how scientists think they're related through evolution. That's why things are put in the groups they're put in. And the way that they come up with this, the way they determine if things are related, is one of the main ways now is they sequence their DNA. So they're looking at like DNA sequences. For example, if we wanted to know how related a panda is to a grizzly bear, they would take a gene, maybe take let's just say the hemoglobin gene from both animals, sequence those, figure out the sequence of A's and T's and G's and C's, and essentially then compare what percent's the same, what percent's different. And the more they're the same, the more they're alike. Not a hard question. The more they're the same, the more the two animals are related through evolution. They also might compare the same thing, sort of, by looking at amino acid sequences. So really that's looking at the same thing, right? Because what determines amino acid sequences? DNA sequences. It's really another way of looking at the same thing. In the current classification system, these are what are called our clades, or our groups. Clade is just a word for a group. So those are our different groups. From biggest all the way down to smallest. Domain all the way down to species. I'm sure in biology you guys had some kind of little saying to help you remember the order. Anybody want to volunteer their little saying? Never heard that one. Is that original?
I think maybe it's in this next class.
Dumb kids playing catch on freeways get smashed. I like it. But anyway, as we start up here, these are what we call the most inclusive groups. These groups are huge. They contain lots of things. When we get down here, these are what we call our least inclusive groups. There's only one kind of thing in the species, obviously. while we're here, just to get an idea of how the system works, how many domains are there in the classification system? 3 domains. A lot of people don't like the way the domain system is set up because there's one domain called eukarya. And what's in that domain? Everything that has eukaryotic cells. So that would be animals, also what else? Plants, keep going. Animals, plants, keep going. What else is eukaryotic? Somebody help me out.
Fungi and protists.
There you go. Fungi and protists. So most of the things that we would think of as important things on Earth, again, that's just from our perspective, fall into that eukarya group. The other 2 groups, though, one of them is called archaea. What's the other one? And really, both of those things are sort of bacteria-like, right? These are our normal old bacteria. What's the EU in front of that word mean? What's the EU in front of any word mean? Like eukaryote. True. True bacteria. These are our everyday bacteria, the ones we talk about a lot. The ones that cause strep throat, and the ones that decompose stuff out of your yard. On the other hand, these are archaea. What's the word archaea mean? I mean, ancient, really old. What's that thing you used to get taught about archaea in biology? Probably in your regular biology class. What did you hear about it? Yeah, but what do they always teach in regular biology class about archaea? Yeah, they're found in extreme environments. I'm going to tell you that's not always true. They're actually probably in this room right now. They're everywhere. It's just that scientists first found them in extreme environments, assumed that was the only place they were at, and they were wrong. They're all over the place. So why are they broken up in their own group? Because they're prokaryotic. They don't have a nucleus. They don't have organelles. Why don't we put them both in the same group? Scientists would argue that these guys right here are sort of the missing link between what? Yeah, between prokaryotes and eukaryotes. Because they have some characteristics that are like bacteria. They have some cellular characteristics that are way more like eukarya. So they have characteristics of both. Another thing we'll talk about while we're here is which groups we belong to as humans. All right, so hopefully, obviously, our domain is eukarya. We have eukaryotic cells. Our cells have a membrane-bound nucleus and organelles, like all other eukaryotes do. Hopefully, pretty obviously, you know you're in the animal kingdom. A few years ago, Matthew, my son, well, he was in like the 3rd grade. He got in an argument with his 3rd grade teacher about classification. She taught the class that humans were not animals. He went in this raging debate for like three weeks with his teacher about animals and humans. And just went on and on. Eventually, I had to send her an email. People are animals, and it kind of ended at that point. But anyway, eukarya, animals. What phylum do you belong to? Chordata. What does that mean? for the vertebrates. The vertebrates. And we'll get more deep. It's actually a little bit more nuanced than that. But for right now, let's just say this is where the vertebrates go. Phylum Chordata. What class do you belong to? Anybody? Full of animal answers today. Mammals, Mammalia. What makes a mammal be a mammal? All right, Jaden said endothermic. That's true. What did you say? Yeah. There's more too. That's actually the main thing.
Yeah, the ability to have like babies for hair usually outside the body.
You're still missing the main one. What does the word mammal actually mean? These are young with like breast milk, breastfeeding. That's what it comes down to. That's what mammal's all about. All right, what about our order? What order do we belong to? That's a word you know. You may not know it. It's the order. We belong to the primate order. What does it mean to be a primate? Yeah, that's the thing that you always hear. Opposable thumbs, right? And you would think, the first thing I said, it's not like a big deal, having thumbs or being. It's a pretty big deal, right? You can grasp things, you can use tools. What other things are primates besides us? Monkeys, lemurs, anything with an opposable thumb. But there's more to it than that. I mean, the reason we have opposable thumbs is because we have very similar legs. DNA. It really comes down more to the DNA than the thumb. We also have three-dimensional vision. All primates' eyes that I can think of are located in the same place ours are, close together on the frame. Why is that sort of important that your eyes are close together? Yeah, I mean, just close one eye, keep your head still, pay attention to what you can see, and then close the other eye and pay attention to what you can see. It's mostly the same, right? The two fields overlap like a Venn diagram. And what that allows you to do is depth. If you got blinded in one eye, even though you can see just fine, are you going to be a very good driver? Nope. You're going to lose your depth perception. You're not going to be you're not going to be able to shoot basketball very well. Probably not going to be able to catch a ball very well anymore because you lose your ability to perceive depth. Anyway, so primates have three-dimensional vision, opposable thumbs. One more thing about the eyes there. Most of the time, if you see an animal that has eyes where ours are at, what does that mean about that animal? Not only does it have three-dimensional depth perception, it's probably where on the food chain? Probably pretty high up. It's probably a predator. Because if you look at things like a lot of the antelope and animals like that on the savannas out in Africa, where are their eyes at? They're over here. What if the animals having eyes over here on the sides? Yeah, you can see 3D, but you can't what? You don't have depth perception. Things with eyes out here usually are prey. They're really low on the food chain. They're not trying to catch food. They're eating grass or something like that. But it's a way for them to see the predators coming and running. That's essentially what that's all about. Anyway, what family do you belong to? This is the one usually nobody knows. It's a word you've heard of before. Sometimes that gets shortened to hominids. What does it mean to be a hominid? It's going to walk upright. Obviously, we're in that group. What else is in that group? Oh, no, not monkeys. What else can walk upright on two legs? Gorillas. Jesus lizards. No, Jesus lizards are not hominids. We're not talking about why, but they can walk upright. Gorillas, humans, orangutans, and chimps. Those are the main things in this group. Now, what other animal do you think a lot of people usually say when I ask this question? Animals that can walk upright. Bears. That's the answer I always get. Never have gotten the Jesus lizard answer before. Can a bear be a hominid? Why not? Because it's not a primate. To be in these groups down here, first you have to be where? You got to be in all the same groups up top. A bear is a eukaryote. A bear is an animal. A bear is a chordate. A bear is a mammal. A bear is not a primate. Therefore, a bear cannot be a hominid. You have to be in the above groups to be in the below groups. Because it's all based on what? Genetics and evolution. You can't skip groups. Jesus lizards cannot be hominids, Draven, because they're not primates or mammals. They're vertebrates. All right, what genus do you belong to? Everybody here should know that. Back up a step. Just homo. Which just means man, in this case. Are there any other things in genus Homo? They're all extinct. But scientists would argue that these would be like the caveman type ancestors of humans. They would have shared this genus with us. Probably Neanderthals and Cro-Magnons and all those other caveman type things. We're the only ones left, and our species is sapiens. And typically when you write out a scientific name, what's it made out of? Genus and species. And there's some rules for writing it. You always write the genus first. You capitalize the first letter. What do you do with the species? When you're writing out a scientific name. You don't capitalize it. And if you're handwriting it like I did, what are you supposed to do to it? You're supposed to underline it to let people know, hey, this is a name, it's a proper name. If you're typing it, what are you supposed to do to it? Put it in italics. So it's genus and then species. If you're handwriting it, underline. If you're typing it, put it in italics. Key things about the classification system. Phylogenetically based today. It's based on evolutionary relationships. It's based on genetic relationships. Things are constantly getting reclassified today as they find out more about their DNA. As they start to sequence more and more things DNA, they figured out they're not what they thought they were. The looks may be deceiving, is what I'm saying. When they start looking at the DNA, they figured out that even though the two things may look alike, they may actually be pretty different. All right, so there's our domains. Archaea, eubacteria, and eukarya. Just probably in the regular biology textbook that you were using, what was usually on the end of the word archaea? It was usually archaea bacteria. They dropped that. It's not the proper name anymore. It's just archaea. They're trying to differentiate them from bacteria. Remember, we said they're sort of like maybe the missing link between these two groups. They have some characteristics of eukaryotes, some of prokaryotes. Remember the whole prokaryotic-eukaryotic distinction. What does it mean to be a prokaryote? Free-floating DNA, not enclosed in a nuclear envelope. You don't have membrane-bound organelles. How much DNA do you have as a bacteria? You have one little loopy chromosome, one little circled chromosome. Whereas eukaryotes, that means true nucleus, have DNA that's enclosed by a nuclear envelope. They have membranes that are also enclosed by, they have organelles rather, that are also enclosed by membranes. They usually have what kind of chromosomes? Linear, not circular. And usually there's a bunch of them, right? Way more than one. What we're going to focus on in the next couple of morning sessions are bacteria mostly, and a little bit about viruses. This is the phylogenetic tree of how scientists think things happen. When we look at a phylogenetic tree, what are we really looking at? What is that? What's that supposed to show you? It's supposed to be like an evolutionary tree of life. And we're going to see phylogenetic trees a lot in this class. What's implied to be way down here? No, not us.
First origin.
The first thing. So first living thing. What's implied to be right there? Not necessarily the second thing. But anytime there's a branch, what's implied to be at the V, at the base of the V?
Speciation.
Yeah, the organism, the common ancestor of the groups that branch off there. So this thing right here is implied to be a common ancestor of bacteria, archaea, and what? Eukaryotes. So if you look at how this thing is drawn, what's implied to be right there? Yeah, common ancestor between archaea and eukaryotes. Notice how the archaea branch is closer to the eukaryotic branch than what is? The bacteria. Scientists would say that these two groups are more related than our group and the bacteria. And we'll talk about why a little bit later on. But again, that's a phylogenetic tree. Remember that wherever there's a V, there's implied to be a common ancestor for the things that come after that V. And typically, the closer together two branches are, that means what? The more closely they're related, genetically and through evolution. All right, the kingdoms in taxonomy. If I asked you the question of how many kingdoms there are before I put up the slide, what would you guys have said? Some people say 6, some say 4. Sometimes I hear 2. Sometimes I hear 7 or 8. How many kingdoms there are in biology depend on which textbook you read, even today. Scientists don't agree. Why don't they agree on how many kingdoms there ought to be?
Subjective.
Yes, it's subjective. They're like, for example, the kingdom Protista, which oftentimes is not considered to be a kingdom anymore. Notice those question marks. It was sort of like the. Well, let me back it. Does everybody have like a junk drawer at their house where you pull it out and stuff that doesn't go anywhere else you throw in that drawer? That's sort of what Protista was for taxonomists. If they couldn't figure out what it was or where it went, they just called it a protist. The things in Kingdom Protista, some of them were animal-like, like paramecium, an amoeba. A lot of them were way more like plants, maybe like a euglena. They could do photosynthesis and stuff. And as scientists started looking at their DNA, what did they find about the DNA of all these things we've been calling protists for years? They weren't really related at all. So there's a lot of debate about whether Protista ought to be considered a kingdom anymore. Your book does not consider it a kingdom anymore. Depends on the textbook you look at. But these are typically the kingdoms that you see in most books. I already did that. So what we're going to talk about today is a little bit about viruses. And this is kind of timely considering what's going on in Africa right now, right? What decision did our government make yesterday about the whole Ebola thing?
Putting on long bracket, sending in troops.
Yeah, we're sending troops to the hot zone. Which in my estimation is a really bad idea, but we'll see. We'll see. Anyway, when we're talking about viruses, the word virus is actually, I think it's Latin, maybe Greek, don't quote me on that, but I think it's Latin. It means poison. And a good way to define a virus is that word right there, that phrase. They are obligate intracellular parasites. Fancy sounding word, simple meaning. What does obligate mean? Not word, the word is obligate, but in this case you say obligate. Still means the same thing as obligate, which means what? Have to. Intracellular means what? Be, not in between, inside a cell. And a parasite means that once it gets in there, it does what? It benefits, and the host cell is hurt, damaged. So it's something that has to be inside a cell, and it causes damage once it gets in there. Viruses themselves are not made out of cells, right? Viruses don't have cells. That's why they're not considered to be what? Living. All right, we're going to start off by talking about maybe the most simple kind of virus, which is called a bacteriophage. Literally, what does that word mean, bacteriophage?
Bacterial virus.
No. Phage. Where have you heard that root at before? P-H-A-G. I know you've heard it in a regular vowel. P-H-A-G. Where have you heard that before? You guys I know have heard of phagocytosis, right? Yeah. What does it mean? Phagocytosis, what does it mean? It's when a cell like engulfs another cell, like eats it. Phage or phago in this case means eating. It means bacteria eater. These are viruses that attack bacteria. Bacteria can get sick. And it's viruses that make them sick. These are viruses that attack bacteria. We'll come back to this slide in a minute. I want us to look at a picture. Here's what a virus like a bacteriophage might look like. All the blue is protein. So most of a virus is made out of protein. And that protein coat around a virus, the outer part, is called the capsid. So this is the protein part of a virus. And then in the middle, usually in the middle, there's some kind of genetic material. And that stuff's in red here. And that genetic material is one or the other. It's either what or what? It's either DNA or RNA, not both. It's one or the other. So these are DNA viruses that we're looking at up here. Anybody heard of this adenovirus thing? This has kind of been in the news lately too. There's a respiratory disease going around in Chicago and St. Louis. Nobody can know for sure where it came from. But little kids, especially kids with asthma, are not able to breathe when they get it. They're going into like the ICU and all kinds of places. There's like an outbreak going on in the U.S. right? And it seems to be caused by this kind of virus. It's a virus they haven't seen in the U.S. before. Kind of scary. Anyway, those are the major structures of a virus. Protein on the outside. Genetic material on the inside. We talked a little bit about in class about these things called retroviruses before. Why is a retrovirus called retro? It's got RNA instead of DNA. And what do we say about RNA? Why retro? Yeah, they think the first genetic material on Earth was probably RNA. Why did we say that's not such a good idea? Yeah, it mutates a lot. It's not very stable. But that's what retroviruses are made out of. They're RNA viruses. Still have the protein coat on the outside, but the genetic material inside is RNA. We talked about this whole central dogma of genetics business. for all living things, that's the way genetics work. DNA to mRNA, that process is called what? DNA used to make mRNA. What's that called? Transcription. Which one are you trying to make up as one? Well, I can't help you if you don't know. Which one are you trying to make up? Cells test, cells quiz. All right. So this is transcription. mRNA to proteins, that is, what's that called? mRNA to protein. Translation. And the little loop thing I'm putting on here, that's supposed to be what? Replication. But retroviruses, they can't do that. They have to have another step. And remember, that other step is their RNA, they inject into that host cell. But what do they inject with that RNA? What else do they inject into you when they infect you? They inject an enzyme, reverse transcriptase. Hold that thought for just a second. All right, so retroviruses, and I lost my writing. They insert their RNA in this enzyme, reverse transcriptase. And that enzyme essentially does the reverse of transcription, which means instead of taking DNA and using it to make RNA, they take RNA and use it to make DNA. We mentioned earlier that a lot of new HIV drugs, probably some of the Ebola drugs, I'm not sure about those, the way they work is they inhibit this enzyme. They block it. And that keeps the virus from being able to replicate. Doesn't hurt you because you don't have what? You don't have that enzyme. You don't need that enzyme. So you're inhibiting a viral enzyme that humans don't have, and therefore you treat the virus without hurting the person. That's the way most antibiotics work as well when we're trying to kill bacteria. Most of them are enzyme inhibitors. But remember that retroviruses, as far as we know, are the only exception to the central dogma of genetics. The whole DNA, the mRNA, the protein, the trait. I'll end today talking about these two things. The lytic cycle and the lysogenic cycle. When you see the letters L, Y, T, we talked about this before, what should you think? L, Y, and T. Yeah, break, cut, tear, split. The lytic cycle is the life cycle of a virus. It looks like this. Ignore the circle part for right now. We'll come back to that in a minute. We're talking about just everything in the main circle. The lytic cycle, the life cycle of a virus. It's in five major steps. The first thing, a virus has to attach to the cell. And it usually attaches to receptors. It doesn't make sense for your cells to have receptors for viruses. You want to have receptors for viruses. So it's not that the receptor is a receptor for a virus. It's that the virus has evolved to use your receptors like what? Like a doorway. So they've evolved to break into your house, your cells, using a door that's already there, using a receptor. So they attach to the receptor. Second step's usually called penetration. And in this step, the virus injects its genetic material. It might be DNA, it might be RNA, depending on the virus. Does the protein part of the virus ever go into your cell? Nope. It's like think of it like a syringe. Protein part, it's like the syringe, where the DNA or the RNA is like the drug. It's the part that gets inserted. Once that genetic material goes into your cells, essentially that DNA becomes part of your DNA, right? And what does your cell do to the viral DNA? It does whatever that viral DNA says, which is pretty much always what? It makes some more viruses. Here's how to do it. So that's step 3, biosynthesis. It copies the viral DNA, it starts building the viral proteins. Your cell does. It becomes a virus factory, essentially. Step 4 is maturation, which essentially, step 3, you're making all the parts. Step 4, you're putting all the parts together. They start assembling the virus pieces. And then typically what happens is that your cells made so many of those viruses that it's overfilled. And what usually happens at the end, they burst out. And that's what we call a release. Kills the cell. Those viruses burst out and they go on to infect hundreds or thousands of other cells. Kind of a gruesome way to go. If you read the hot zone, you know that in Ebola, you can actually see that, right? What does it look like in the hot zone? What do the virus pieces look like? Because they're so concentrated. You know what I'm talking about? When they talk about like how they vomit, what's in the vomit? When they bleed, what's in the blood? Black specks. Those black specks are viruses. Millions of them. And as far as we know right now, this Ebola outbreak that's going on now, how's it being transmitted? It's being fluid contact. So blood to blood, blood to like eye, blood to mouth, blood to nose, fluid contact, as far as we know. Because these fluids from these Ebola patients they're like leaking viruses essentially, right? And in Africa, it probably wouldn't be as bad in the United States. Part of it's because of the burial rituals and the way they treat dead bodies over there. There's a lot of places in Africa, the customs are like kiss on, hug on dead bodies. Don't tend to do that much over here, right? So I don't think it would be transmitted as well over here as it is over there, because it seems to be right now, if the government's telling us everything, and I hope they are, that it's being transmitted by fluid contact and not like some airborne version. If that's the case, that would be bad, right? Hopefully we're not there yet. All right, the last thing I'll mention for today. Some viruses will sort of take a time out from the lytic cycle. And their DNA will become part of the host DNA, and it'll just sort of sit there, sometimes for months, sometimes for years, and cause no symptoms at all. So the DNA gets injected, and it just becomes part of the host cell's DNA. So for example, right here, the blue, that's the host DNA. The red is the virus DNA. They're connected. When it's in that situation, it's called prophage. It's virus DNA that's connected up to the host DNA. Prophage DNA. And like we said, there's no symptoms going on during the cycle. It's just dormant. But something can cause that to change. What's an example of a human virus infection that works sort of like this?
Rabies?
I don't think so with rabies. Rabies, it takes a while to get the symptoms, but I think it's more that it's just building up enough viruses to cause the symptoms. What I was thinking about shingles. Most of you, well, maybe not you guys, a little older people, how many of you had chickenpox? Let's just ask it this way. Actually got the disease chickenpox. 2 or 3. Used to, if I'd asked this 10 years ago, if I'd asked that question, everybody in here would have raised their hand. Why has that changed? Now they have a vaccination that they're starting to require people to take. But 10 years ago, that vaccination didn't even exist. Once you get the chickenpox virus, eventually you get over chickenpox. chickenpox. You have a few bumps, you itch a little bit, you get a fever, you don't feel good for a day or two. But typically, it's not a serious infection. But once the symptoms go away, is the virus gone? It's still in you. So Justin still has chickenpox inside him. And he always will. And later, if something goes on with his body, like maybe something causes his immune system to get weak, maybe he gets all stressed out at work, maybe he gets sick with something else. A lot of people who get shingles end up finding out later that there was something else wrong. Maybe they had cancer, maybe they had HIV, maybe they had hepatitis, because that other disease has caused their immune system to not work the way it's supposed to work. And it comes back. So that same virus that caused chickenpox later causes shingles. It's the same infection. If Justin had shingles and he was around a kid who hadn't been vaccinated, what's the kid going to get? The kid's going to get chickenpox. So a person with shingles can give somebody who's never had chickenpox. It's the same virus. There are several viruses that can do that. They just sort of get in your body, remain dormant, and then they show up later. They thought that's what AIDS was doing for a long time, because typically, what do people with HIV, how long does it take somebody who gets infected with HIV to get symptoms with AIDS? Way longer than that. Like 8 to 10 years sometimes after infection before symptoms show up. They thought this was what was happening, but now they're starting to decide that maybe your immune system's been fighting. It's been trying. And it just eventually loses. Can't win. But it's fought them off. It's fought off the enemy for 8 to 10 years and just eventually gives up and loses the battle. But the lytic cycle, I'll end with this. The lytic cycle is the normal life cycle of a virus. Virus attaches to a receptor, injects its DNA or RNA. The cell becomes a virus factory, starts making virus pieces. Those pieces get put together, and finally, the cell bursts, and out come the pieces. And remember, the lysogenic cycle is sort of when the virus takes the time out from that. It injects its DNA, it becomes part of the host DNA, it's like a prophage, and it remains there inactive, sometimes for years. It's a good place to stop. Before you go, is everybody on the list? Anybody who did not put their name on the list? All right, good place to stop.